Right, guys? Please follow the instructions that are right now on the screen. If you don't remember how to do any of those parts, please don't hesitate to ask either David, Juan, or myself, or Reinhard. First thing, open your VirtualBox and start your BioLinux 8 machine. That's the first thing. So the first steps are this one. First, open the VirtualBox. Okay, so first open the VirtualBox. Then you'll already have 1, 2, or 3 different sessions of BioLinux open here. Just double click one of them. So to create your shared directory, you go to Devices, Shared Directories, Preferences. Please everybody, if you haven't done this part, you can follow these instructions here. Go to Devices, Shared Directories, and Preferences of Shared Directories. Here. Has everybody done this already? Yep. Cool. Here you can see I already have it set up, but it's not mounted anywhere. In Documents, we're going to create a directory called INEA05. Can you guys see that? Yeah, just create a directory under Documents. Please make sure that your path, your full path, Home Manager Documents INEA05. That's going to help us so that everybody has the same kind of environment. That way we know where each of you have the files that we'll need. Everything? No. The only thing you need to remember is that you need, at this point, you need to create a directory called under Home Manager Documents, a directory called INEA05. Yeah, and you also need to create a shared directory using the dispositivos. Okay, guys. Again, please follow these instructions. Once you open your machine, once you start your machine, please go to Devices or Dispositivos. Go to Shared Directories, Carpetas Compartidas, and Preferences o Preferencias de Carpetas Compartidas. You need to, if you don't have this this directory like this, yeah, you don't have anything but these two lines, carpetas de la máquina y carpetas transitorias, please press this button, Add New Shared Directory. Then you'll get this dialog box, yeah, and where it says Path to Directory, you look for another one. Yeah, and here you go to directory INEA05, the one you just copied from the USBs. You just go there and select it, like this. I know it's in my documents, I know it's in personnel, here, and here it is. So once you go to wherever you copied the directory INEA05, you put select directory. That's all you need to do. Then you just have to do, yeah, and please make it permanent so that you don't have to do this every time. So is everybody on this dialog table already? Have you been able to create your own shared directory? Please be aware of what I'm putting. I'm not putting any mounting point. Punto de montaje queda vacío. No lo vamos a poner hoy día. Just select auto mount and also make it permanent. All right? And also, do not create another directory inside of what I've already given you, inside of INEA05. Just select INEA05. Yes. So far, so good. I just want to make sure that everybody is on this step at the moment. So everybody's already here, right? Everybody has already created their share folder. Yeah. So are we all on the same page now? Has this been done? So now that you have done this, you have created your shared directory. Yeah. The next step is to create the point where you're going to mount it. Yeah, I've asked you to create that INEA05 directory under Home Manager Documents. This is in your Linux machine, in your BioLinux machine. Go CD into Home Manager Documents and create INEA05. If you don't remember the command to create a directory, it's mkdir, make directory, mkdir. D-I-R. Now you can either CD into this new directory or you can just stay out of it. It really doesn't make much difference for the next step because the next step is to manually mount it. Yep. manually mounted. So please remember this direction. We all have the same direction. /home, /manager, /documents, /INEA05. And once you have that, what we're going to do is we're going to do this. Please let me know if you can see that command I just wrote. It's starting in sudo, S-U-D-O. Can you see it? Is it clear? Does it need to be bigger? If after you finish writing that, you press enter, and now you list the contents of the file of the directory INEA05 from Linux, you should see all the files that you had on your Windows folder in there. Please be aware that we're talking about two different paths. You have one path in Windows, your host machine, yeah, that it's wherever you put the INEA folder when you copy it over, yeah, and then I'm asking you to create a different path, a different directory inside of Linux, inside of your virtual machine. And the directory I'm asking you to create from Linux is /home, /manager, /documents, /INEA05. Yeah, what we're trying to do right now with all these commands is to link the directory in Windows with the directory you just created in Linux. We are trying to link them. Okay, guys, I just want to make sure that most of you are already there. Yeah, we're going to start moving forward. There are just a few machines that are not working properly. Hopefully we'll solve that pretty soon. But in the meantime, we'll just keep moving. So basically what you've got here, yeah, it's 4 main files. The first 2 are reads. It's what you would get if you get something sequenced on an Illumina machine. This is what you get. Yeah, if you want to take a look at what it looks like when you get a sequencing result from a sequencing provider, yeah. Do you see those files that said sim _1.fastq.gz? Yeah, those are what we call FASTQ files. Those are sequences. Is everyone familiar with the FASTQ format? Yes? It's similar to FASTA. It has more information. But is everybody familiar with the FASTQ format? Let's take a quick look. We're going to use something we learned the first class, piping. All right? So the first thing you need to understand here is that from the extensions, and from the extensions alone, you have a gzip file. That means that if you try to open it with something like, or if you want to try to visualize it with something like head, you cannot see it directly because it's zipped. That means it's compressed. There is a very quick way to unzip it. The command setcat. Setcat is a command that basically will uncompress and will print it on the screen. I'll just give you a quick look at it. Yeah, and I'm going to interrupt it because it's not going to finish in forever. All right? It just keeps going like that. So we don't want to see the whole thing. We just want to see a snippet, like last time. Yeah, so instead of just doing this, yeah, we pipe it, and we just want to see the head. Does that make sense? Yeah? And what's more, I want to see the first 20 lines. Just the first 20 lines. Does that make sense so far? Does the command that you're seeing right now make sense? Let me just put it like this, so that you can all see it. So look at this command. It starts in setcat. Setcat is to uncompress and print at the same time. Yeah, it decompresses and prints. Huh? Yeah. That interrupted. If you want to interrupt it, just Control C. If something you don't want it to happen, it starts happening in your screen, just Control C will stop it. Control C will finish. All right. So just take a look at the command I wrote on the screen. Setcat. The name of the file. You can choose either that file or the other sim _2.fastq.gz. Yeah, and then instead of pressing enter, you pipe it to a head -n20, which means I only want to see the first 20 lines. All right? Now you press enter, and you'll have a more sane result. It probably doesn't make much sense yet, but you kind of understand that there is some sequencing there, right? Even if you're not familiar with FASTQ format, you do realize that there is some sequencing there. So let's just take a look at this and realize that there is a pattern. There is a plus sign along every 4 lines, right? Every 4 lines, you'll see that there is a plus sign. And that's the only thing that will appear in the line. Yeah, that is because the FASTQ format, yeah, basically it's a way of writing down sequences, yeah, that occupies 4 lines per record. 4 lines per record. The first line, as you can see here, it's the name. And how do we know it's the name? Yeah, because the format, just by convention, it's just something that people decided, oh yeah, let's just agree to this. They say, okay, the @ symbol, la arroba, the @ symbol, is telling me, okay, this is where the name starts. The first name is SIMSEC number 1, or SIMSEC _1. The second line gives me the actual sequence. CAAGGATT, and so and on it goes. The third line, it just has a plus sign. It's a separator. Yeah, it's just it's kind of like a blank line. It doesn't really serve any purpose. Yeah, and the fourth line, you will see that it's a mix of different symbols, numbers, characters. Yeah, it looks like rubbish. Yeah, but this is actually a way that Illumina uses, yeah, and most among people who use FASTQ uses, yeah, to codify the quality of each of the bases. For each base that you have on the second line, you will have a character on the fourth line. So the second and the fourth line will always be the same length. They'll have the same number of characters. Why? Because for each nucleotide that you have on the second line, you will have an associated quality value. Now, what is a quality value? Of course, if it's higher, it's better, right? I think we can all agree to that. But how does quality is measured in this sequencing? The quality is a direct relationship with the probability of being a mistake, being a miscalled base. Yeah. For example, let's assume that you're not looking at just random characters, but you're looking at numbers. Yeah. So, Reinhard, could you check what's the ASCII value for capital E? The ASCII value for capital E. For capital E. So basically, there's a transformation from the ASCII value of each of these letters. Yeah, there's a transformation, and you get a number. Yeah, that number is the negative logarithm of the probability of this base being wrong. It sounds weird, but I'll make it simple. If something has a quality value of 10, that means that the probability of it being wrong, yeah, it's 1 in 10. So that would translate into every 10 bases, you probably have 1 error. So to speak, yeah. 33, the E? 73? I am minus 33 would be 40. Okay. So if something has a value of 20, yeah, that basically means that the chance of it being wrong is 1 in 100. If something has the value of 30, the chances of it being wrong is 1 in 1,000. If you start making the correlation, you'll see that it's not the 0 that really matters, it's the number after, to the left of the 0. If it has a 40, then it has 1 divided by 10 to the 4th, which will be 1 in 10,000. If it has 50, it is 1 divided between 10 to the 5th, 1 in 100,000, and so it goes. Yeah, so basically that's what's being encoded here. This is a FASTQ file. Yeah, and FASTQ files are usually what we call multi-FASTQ files. We have hundreds of thousands of records in each of these FASTQ files. Hundreds of thousands. Yeah, if not millions. This one, if you want to see the size of these files, just do the command "L" and enter. And you will see the size in bytes. Basically, this one is 99 megabytes. It's really small. Now, could any of you tell me how many sequences we've got? Would any of you just try to guess? How do we know? How could we know how many sequences there are in just sim1.fastq.gz? Using the things that we already know. Does anybody remember how to count lines? Okay, the command to count lines is WC space minus L. It's word count space minus L. We're telling minus L because we're telling we don't want you to count words. We want you to count lines. So what we do is pretty much the same we just did. We pipe it. First we do secat sim1, and then we pipe it to word count minus L. If you cannot see it, I'll just, something in your configuration has changed. So it's better just to clean up, restore it from fresh, remount it, yeah. And has everybody already checked this command and press enter? How many lines do you have? 4 million. You have 4 million lines. Every record has 4 lines. How many sequences do you have? Now, there is a reason why sim1 and sim2, they both have the same number of sequences, the same number of lines, and therefore the same number of sequences. It's because these are part of a library construction called paired end libraries. That means for each template DNA that you have to sequence, you sequence it from both sides, from forward and from reverse. So the sequences that you find in sim1, they belong to the forward sequencing. The sequences you have in the sim2, they belong to the reverse sequencing. Yeah, there is an entire statistical approach to what is the average distance between the two sequences in the real world. Yeah, because you selected the size that you wanted to sequence, and also by selecting the size, you set up what is the standard deviation. Yeah, so if you aim for around 300 base pair as an insert size, yeah, then you probably have plus minus 100 base pairs. So you have between 200 and 400 base pair insert sizes. And there's going to be pretty much a normal distribution of the sizes, of the distances between each of these pairs. But that's something we cannot see from here. We can only see the sequences. To actually look at that, we will need to either assemble it or map it to a reference genome. So now that we are here, yeah, now that you've already seen what a FASTQ file looks like, how to open it, how to explore it a little bit, yeah, there are many more ways to explore it, yeah, but you've done the first quick, easy part, yeah. Then now we can start the theoretical/practical session, genome assembly. All right. So de novo genome assembly. This is the subject for today. Does anybody have like an idea, has ever worked on this, or has any knowledge about this that you would like to share? Have you ever heard this term? The term de novo genome assembly. Have you ever heard it? Yes. Do you know what it is what it stands for? Yeah, no, that's a very good start. So basically, when we talk about the Novo genome assembly, we're talking about putting back a genome in order, like inferring the original sequence of a whole genome. What is the order? The presentation today is going to take several parts. The first part is we're going to talk about what is an actual de novo genome assembly. Then we're going to talk about what is it useful for and what it's not useful for. We need to have that very clear. Then we're going to talk about the sequencing technologies available and the sequencing approaches available to tackle a whole genome assembly. Then we're going to talk about the assembly algorithms that are the mainstream to assemble and to infer the sequence from those small pieces of sequences that we're getting. Finally, we're going to see step by step what are the tools and what are the methods and what's the reasoning behind all the steps for genome assembly, from the pre-processing and just data exploration part of our reads, all the way to the assessment and validation of the reference genome that you just built. All right? We have a small test, a very small test, that you will run your own genome assembly on your laptops, if you have enough memory, if it doesn't crash. Yeah, but it's a very, very small genome. Yeah? Those reads that you just were exploring, SIM1 and SIM2, yeah, they belong to a genome. Yeah, and we want to see if you can infer the original sequence of it and how much it looks like the actual reference genome it was based on. And finally, once we see the essential steps, we'll do some final remarks about how each species that you tackle, that you want to do the Novo genome assembly, it's different, it's going to have its own problems, and it's going to have its own methods of tackling those problems. So what is a de novo genome assembly? A de novo genome assembly is basically a process, or the process, of reconstructing the most likely original sequence of a genome. And from what? We're reconstructing it from small overlapping pieces, small overlapping fragments that we call reads. Regardless of the technology you use, yeah, whether you use Illumina, PacBio, Nanopore, all of these we're going to produce fragments that are smaller than the original DNA molecules we're trying to reconstruct. So the reconstruction is going to be like solving a puzzle. That's pretty much the same equivalence everybody uses, and I think it's an appropriate equivalence. Yeah, it's like solving a puzzle. Now, it is one of the hardest problems in bioinformatics to solve, and yet there are thousands of tools dedicated to just tackling this problem. Thousands. Each of the tools that are called, well, for the main part that are the assemblers, yeah, they have their own pros and cons. They have, some of them are just accept one kind of library, some of them can only use a combination of different libraries, some of them can use just one sequencing technology, some of them can tackle many different sequencing technologies, some of them are only dedicated to bacterial-sized genomes, some of them are dedicated to mammalian-sized genomes, some of them can handle very repetitive genomes, some of them cannot handle very repetitive genomes, and they crash. Some of them use a lot of memory, some of them have optimized their algorithm to reduce the memory footprint as much as possible. Some of them are multi-threaded, so they will run faster, and some of them are not multi-threaded, they are single-threaded, which means they can only use one CPU at a time, and therefore they are slower. So all of these variations and these, these, let's call it games, it's what you're going to find when you start thinking about trying to tackle your first de novo genome assembly. And trying to decide what is the right tool for you, it's usually one of the most difficult areas. Everybody likes to recommend their own assembler. I have my favorite assemblers because I tested a few. Yeah, and when I talk to another bioinformatician, he uses a completely different set of tools for every single step. They are all useful. Yeah, it just comes down to personal preference. It is costly, yes. Even though the prices of sequencing have dropped, for us who work in plant genomes, we know that plant genomes are usually way bigger than most bacterial and mammalian genomes. So plant genomes are usually a lot more expensive because you need to sequence much, much more data than what you would need for just a bacteria, parasite, or even a human. Now, yes. Oh, it could be huge. It could be huge. For example, last big genome I worked on was the wheat extrapolate genome. The expected genome size was 17,000 million. That's 17 billion base pairs. That is 5 times the size, well, almost 6 times the size of the human genome. Yep. And the original paper of the assembly had an average contig size after assembly, yeah, of around 1,000 base pairs. Wheat, yeah, 1,000 base, yeah, that was. I don't know if you're familiar. We're going to talk about metrics, but N50 means the size of the contig above which, yeah, half of the genome has been assembled. So, for example, if we're talking about a 17 gig genome, half of the genome would be 8.5. Yeah, right? 8.5. So that means that the size of the genome, if you start summing up the longest contigs until you reach 8.5, that will be your N50. And that's an indication of how contiguous your assembly is. For wheat, the original one was 2,000 base pairs. It was really poor. It was really poor. And why? Because wheat is one of the most repetitive genomes that ever existed. It's between 75 and 85% of the genome is considered repetitive. Yes, and these repeats are not small and are not simple. They are complex repetitions that are nested one within the other, and you have paralogs and you have orthologs. It's a very messy genome. Yeah, the statistics were horrible. Yeah, and then just 2 months later, they developed a new assembler that was specifically designed to tackle the wheat genome, the repeat level of the wheat genome. Yeah, and the assembly was just something completely different. Average contig size of around 10 megs, if I remember correctly, so over 10 times bigger. The N50 was nearly half a gig. Yep. And then when they started including more and more libraries with different sizes and adding some more technology, they got nearly chromosome scale assembly. So one single contig that was containing a whole chromosome in one single contiguous sequence, which is something that it's just becoming possible right now. It wasn't possible before. So yeah, the decision of what assembler to use, what tools to use, can make a huge difference on the quality of the assembly you have in the end. That's where background research comes in. You have to understand what are the characteristics of the genome you're trying to tackle. If you know that your genome is not very repetitive, then you do not need an assembler that is specialized on repetitive genomes. If you know that your genome is not too big, then you do not need an assembler that will tackle huge genomes. If you know that your genome is big, then you do need that kind of assembler. So you need to have an idea of what you're dealing with before you embark in a de novo genome assembly. Now, there are two main approaches to de novo genome assembly. Yeah, there is the hierarchical approach, and there is the whole genome shotgun approach. I'm going to touch what these mean. It requires a little bit of molecular biology background. Yeah, but the idea is very simple. In both cases, the technology doesn't allow me to sequence a whole chromosome in a single go. In neither of both of those approaches. So the first guys who decided to go for genome assembly, they decided, okay, let's do it the simple way. Let's create a back library. Yeah, we don't know the order of the backs within each other, but we're going to use restriction mapping, restriction patterns, to more or less create a path. What's the name of the path? FTP path? No. Well, doesn't matter. So let's create a path. So let's sort the backs without knowing the sequence that's inside of them. Let's sort the backs based on how similar their restriction patterns are. They just took a bunch of back sequences or back clones. They put a series of restriction enzymes. They look at the restriction fragments, the sizes, and they said, okay, these are the fragments. So whichever two backs started sharing the same fragments, they would say, all right, these two backs that share many fragments using the same enzyme, they are probably overlapping. And since they are probably overlapping, they should be contiguous one to each other. Yes? Oh, bacterial artificial chromosome. It is a method where you can just chop a genome in big chunks, let's say 100 kb per fragment, and then you clone it into a bacterial artificial chromosome, and you keep it in a bacterial culture. It is stable. You can keep it for many generations. You can freeze it, and when you take it back, you still have the same bacteria. Each bacteria will have a different piece of the genome. So they create first this let's call it back path, a path. It's kind of a golden path. You know what back is contiguous to the next one. It's contiguous to the next one. It's contiguous to the next one. So after you do know that, then you say, okay, let's tackle each back at a time. That's why it's hierarchical. Let's do it from the top to the bottom. Now let's sequence the first back in the line here, the first one here. How do we do it? Because it's still way too big to sequence it alone. You have several approaches, but basically what they first did was they took that piece of DNA from one single back, 100 kb more or less, they fragmented it, and they subcloned it. They subcloned it into smaller backs, or into smaller vectors, also bacterial vectors. And then each of these smaller bacterial vectors was going to be sequenced. In that time, they were going to use only Sanger sequencing. That's how the original idea of the human genome sequencing came to be. They wanted to use, and they actually finished using this approach to do the human genome. And in the last years, there actually was a competition between this approach, which was predominant from the 80s all the way to the end of the 90s, and the new approach that appeared just in the early 2000s, the whole genome shotgun. The whole genome shotgun, the idea of it is that let's not clone nothing. Let's don't clone nothing. The only thing we're going to do is we're going to chop all the DNA in sequences small enough that we can sequence them all in parallel. And why did this happen? Because new technology arrived. Massively parallel sequencing. Massively parallel sequencing in the form of, I don't remember the name of the firm. Do you remember the name of the first machine? It wasn't Illumina. It was another name. Well, doesn't matter. Yeah, but the company that generated this first machine, in the end became Illumina. Then a lot of other technologies appear later on, 454, ion torrent, etc. Yeah, but the idea was always the same from that point forward. It's like we are going to sequence massive amounts of DNA all at the same time, all in parallel, so that we can get enough coverage of the genome in one single go. We don't have to do this hierarchical thing anymore. But with that kind of technology arose another problem, is that here, at least you know what back goes after the other. what's the order of the bags. And therefore, you kind of have an idea of what's the order of the sequences of these bags once you assemble, once you get their sequences. You know that they are overlapping, so there must be some overlapping threshold, and then the sequences should be similar in some parts, so you can just keep building one after the other. Yeah, but here, when you fragment the DNA and you sequence it all at the same time, you have no idea where each of the sequences you're getting belongs in the genome. It's a mess. You have absolutely no idea. And that's where the first approach to tackle these kind of sequences, this kind of massively parallel sequencing came to be, the deep bridging graph. We're going to talk about later on, yeah, but this new sequencing technology required a new algorithm for tackling the assembly problem. We couldn't rely on the hierarchical sequencing anymore, or because we weren't using the same technology. We're using new technology that had different characteristics, yeah, that made it impossible to know or to guess or to know beforehand which sequence was contiguous to the next one. Now, why is genome assembly useful? When should you use it? First, it is useful because it provides a physical map of an individual's genome. That is a huge step. Many times we're using markers that we don't know where they stand. Many times we've already related a specific marker with a specific phenotype, and we know that this marker will tell me if this plant is going to die or is going to survive this condition I'm going to put. But I have absolutely no idea, we have absolutely no idea what genes are around it, or what makes this marker so special that it confers or it signals that the phenotype we want is present. That's usually what happens if you use AFLPs, rapids, etc. That's usually what happens. When you have a genome assembled, yeah, you can find the sequence of your marker, and then you can find what is the surrounding sequencing. That doesn't mean that's going to tell you a lot. There's still a lot of work after that. But still, you know the context in which the marker you found where it is located in the genome. So it offers an anchoring space for previously unplaced markers. SNP tags, SSRs, AFLPs, CAPs, all these sequences, you can finally anchor them. You can finally see the relationship between the genetic maps that you had created using hundreds of markers, yeah, and the physical distance and orientation of those in the sequence. It enables the discovery of novel genes. This is combined with other technologies, annotation algorithms. Yeah, but yes, it enables the discovery of novel genes. It facilitates synthetic studies. You can compare the order of the genes or the order of the sequences between different individuals or between different species. And you can relate that to their evolutionary relationships. Also, it increases the scope and resolution of those synthetic approaches. Also, it enables the discovery of higher density molecular markers. Since you have the sequence already inferred, yeah, you have a lot of DNA where you can design new markers. Markers specific to whatever you want. Now, what it's not useful for? It will not give you on its own information about the function of different regions. By itself, it will not give you that information. You can use transcriptional data, you can use interaction data to make guesses or inform guesses about what each part of the genome is for. You can use some basic statistics, like an exploratory analysis about GC content in windows of 1,000 base pair, and guess, yes, these are GC rich content, these are GC poor regions, but yeah, that doesn't tell you anything about the function of that. That only tells you, oh, this is a characteristic of it. Maybe it could be related to something special, like methylation, higher transcription rates, or lower transcription rates. But you need additional information for that. It requires additional information to understand its organization into coding and non-coding sequences. This goes back to transcriptomics. It is not enough to prove if it's the cause of a phenotype. Just because you have what appears to be a gene, because you've done some ab initio gene prediction and found that there is a gene and it looks like something that has already been found in the database, yeah, it doesn't mean that it's being transcribed, and therefore it doesn't mean that it's affecting a phenotype. It could be a pseudogene. It could be, well, there are many options there. And finally, a genome assembly will usually be incomplete. What do we mean by incomplete? It's going to have a lot of gaps. Even the human genome, which is one of the most refined and studied genome, the genome where more people have worked on, it has a lot of gaps. It has a lot of context that hasn't been placed to an actual chromosome yet. Of course, every year the number of gaps is reduced, and we have newer and newer versions of genomes. Yeah, but so far there is not a single genome that doesn't have gaps. Well, maybe some virus, some bacteria, but other than that, the plants that we're working with, they are all going to have gaps. So, what are we going to use to sequence? Once we decided the approach that we're going to use for sequencing, and let's assume for the sake of argument that we're all decided that it's cheaper to just do whole genome shotgun, yeah, we have to decide what technology we're going to use for sequencing. And it depends, the technology we're going to use, it depends on two things: the complexity of the genome we're trying to tackle and our budget. So the first technology is sequencing by synthesis, the Illumina technology. Basically, well, we haven't touched the library preparation in this spot, but basically you fragment the genome, usually through sonication. Yeah, you get, when you run a quality gel of what you've destroyed, or how you've destroyed the DNA, you usually see a smear. You're familiar with the smears, right? All right, so you see a smear, and from that smear you select a certain size range. The size range depends also on how repetitive you expect the genome to be. Why? I'm going to explain that later in algorithm, but basically it's the smaller the insert site, the smaller the template, the more difficult it's going to be to tackle repeats, to solve the repeats. And therefore, the repeats are going to be collapsed, and the assembly is going to be more fragmented. Yeah, we're going to see that when we see the algorithms. Yeah, but basically, first you choose an insert size. After you choose the insert size, you start the library preparation, and the library preparation basically means that you polish, you add some adapters, you polish the adapters, you add some priming sequences, and then you just put each of the sequences, you make sure that each of the sequences are attached to a bit. The bits are going into a chip or into a flow cell that is going to go into a machine. But before that, you do amplification, because what they're going to do is they're going to start adding one base at a time in hundreds of thousands of bits that are very close to each other. And every time one base is added, a little bit, a different light, a different wavelength of light, or different color is going to be produced. If you have only one sequence per bit, yeah, the amount of light that is going to be produced, it's not going to be enough. So what you do is you clone in the same bit, you clone over and over and over until you have a good, nice population of clones of the same sequence. So each bit will have a population of clones of the same original sequence. So that every time you add one nucleotide to each of those clones, you will not have just one small light. You will have a big light or a bigger light. And that can be captured by cameras. that is basically what we're doing when we do sequencing by synthesis. Is that every nucleotide that is added will emit a color, yeah, and there is going to be a camera, a high-speed camera, that is filming the process of adding each of the nucleotides in every round. As that happens, yeah, each of the bits will have a different population of reads inside of it. Yeah, so each of the bits will emit a different color in each round. So when you see the film, it's going to be like a small square with a lot of spots, bright spots of different colors, just turning on and off, on and off, on and off, over and over. That's the actual raw data that you get from sequencing by synthesis. Now, what's the next step? Is to convert that video of flashing colors, yeah, into guesses of what has been added. If we are used to saying that red codes for adenine, yeah, then we say every time we see a red flash in one spot, that means that the sequence in that spot has just added an adenine. So therefore, we can add an adenine to that position. There is a lot of error to go around. You can mix up the colors from bits that are very close to each other, or the colors that are close to each other may just camouflage each other. The machine can just make the error and not understand the coordinates where it's reading the light correctly, and you start reading from one coordinate and the other coordinate at the same time, which can also be tricky. So each of those possible errors is going to be taken into account, yeah, when guessing back the sequence. And that's where the quality score comes from. The quality score that we mentioned before, the probability of being the wrong base, comes from those errors that are taken into account. If at the time that the base was added, there was a lot of light around that area of the flow cell, yeah, then the probability of it being wrong increases, and therefore the quality decreases. All these things are taken into account by the machine, and they have specific algorithms to do that base calling. This is the process called base calling, trying to convert these videos into actual FASTQ data, the one that you just saw. Luckily for us, yeah, Illumina has done an amazing job in improving their base calling algorithms, and more often than not, you get high quality sequence from Illumina machines. Yeah, the most frequent reason for low quality libraries or for low quality sequences in Illumina machine is poor management of the machine or a bad library in the lab. That's usually the case. Single molecule real-time sequencing, the smart sequencing. This is technology used in PacBio. Yeah. This produces, I forgot to mention this one. So the first machines in Illumina, they only produce reads the size of 32 base pairs. 3 and 2, 32 base pairs. And the first genome that they attempted to assemble it using only fragments of 32 base pairs. Imagine how difficult it would be to tackle a human genome, which is 3 billion base pairs, using 32 base pair reads. It is a huge challenge. But right now, you can find machines that can produce reads up to 300 base pairs. I don't know if they've gone any further, I don't think so. But yeah, up to 300 base pairs. But that's their limit. There are optical limits and physical limits to how much the sequences using this sequencing by synthesis technology can achieve. You cannot go any bigger, because the bigger the template that you're sequencing, the more likely it's the color is gonna be read from a different coordinate in the flow cell, and if you're reading it from a different coordinate, there is a higher chance that it's going to be erroneous because it's going to be crossing with another bit in the same flow zone. Single molecule real-time sequencing. Basically, they developed also a whole new technology to do the same thing. It's kind of like sequencing by synthesis, because basically you have a polymerase that will attach to one strand of the DNA and will incorporate nucleotides. And each nucleotide, at the time of being added, is going to generate a small light. Yeah, but in this case, they've avoided altogether the cloning or the PCR step, where you have one single copy per bit and you increase it into a whole clonal population of reads. They don't do that. They just keep the one read per bit, yeah, and they've generated very sensitive zero wavelength pockets. Oh, a special reading machine that can see the incorporation of each nucleotide in real time for just one single molecule. That's why it's called single molecule real-time sequencing. Because they can read, they basically are seeing how each nucleotide is being added in real time. And in real time, they're saying, okay, this sequence is being added to this well, yeah, so we can add that sequence into, or that nucleotide into the sequence of that template that we have been sequencing there. Also, let's talk a little bit about the library. The library is what they call a smart bell or double bell library. It's just a single-stranded DNA. Well, it's going to be a single-stranded DNA that has two hairpins in the end. Where is that? Oh, there we go. Yeah, and when the polymerase attaches, is attached to it, it's going to start going in circles. It's going to start here. It's going to just use first this line as template. It's going to go through the other loop. It's going to take the other side as template. And therefore, it's going to go rounds and rounds and rounds. Even though this area here comes from an originally double-stranded DNA, yeah, they've made these adapters, these hairpins, in such a way that during sequencing, each of the strands of the original template is going to be sequenced as if it was independent. Yeah, so you're going to have what they call circular subreads. The polymerase is going to read from the hairpin. It's going to go through the yellow strand, the plaza strand, let's call it. It's going to take another round through the hairpin. Then it's going to sequence the purple, which is the complement of the yellow one. Then again the hairpin, then the yellow, the hairpin, the purple, and so on, so on, so it goes. Yep. The average time that a polymerase remains attached to this nucleotide, it's around 20,000 base pairs. So if the insert size is just 5,000 base pairs, yeah, and the polymerase remains attached to the DNA for 20,000, it's going to go around it 4 times, more or less. So that means that for each template, you're going to have more or less 4 subreads. That's what they call here as subreads. You have forward, reverse, that's it. That's as much as it got. Now, the most recent technology or the most recent release of a polymerase of a chemistry from PacBio, they say that they can keep the polymerase attached for nearly 100 kb. So that means that for these smaller molecule fragments, you can get a lot more sequencing and therefore a lot less errors. But most of the researchers are not going to use it for this so-called circular consensus sequencing. What they're going to try to do is they're going to try to increase the size of the template. It doesn't really matter if you get 2 or 3 rounds of each read. What you really want to get from this sequencing is long-range information. You want to have longer reads. What else can we say about this? Well, the fact that it has a much larger range of reads. If with Illumina we're talking about 300 base pairs maximum, here we're talking about a minimum of 3 kb, 3,000 base pairs. And lately, the average read length, yeah, is reaching 15,000 base pairs. So it's really big. Now, there are some caveats regarding this technology. The first one is that you need high molecular weight DNA. What do I mean by high molecular weight? That most standard DNA extraction methods that are designed to target just small fragments of 1 kb for amplification or microsatellites, they don't really care that much about high fragmentation of the DNA during the extraction process. Here, if you want to really take advantage of the smart sequencing technology, you need to be able to obtain DNA that has an average size, an average weight of around maybe 20,000 base pairs. That's what you're aiming for, and that's not easy to accomplish. And that takes us to the next one, nanopore sequencing. Nanopore sequencing, it's basically the use of nanopores that are just enough, they have enough space to allow one strand of DNA to go through the nanopore. Yeah? And the machine, what it does, it reads the changes in electrical potential in the membrane where the nanopore is. The hypothesis behind it is that each nucleotide will cause a different change in the electrical potential of the membrane, and therefore each one will have its own signature. And by its own signature, it means that it shouldn't be too difficult to differentiate when a C is being added or when a C is going through the pore compared to when an A is going through the pore. But as a matter of fact, it is a lot more difficult than they originally expected. And that is part of the reason why this sequencing technology hasn't taken off as much as it could have or as much as it promised, because the error rates of this sequencing technology are skyrocketing. From personal experience, they promise that they have around 10% error rate. That's a promise. The reality is they have around 25%. So that means that 1 every 4 nucleotides, it's probably wrong. That's huge. Now, they try to compensate this the same way they do it with single, well, I forgot to tell you, this also has much worse quality than Illumina sequencing. Whereas Illumina sequencing, you have an average of 1 error every 1,000 base pairs. This one has, in reality, 1 error every 15 base pairs. So it's big. It's a lot of sequencing error. And what they propose to reduce the error rate is to increase the coverage per sequencing experiment. So that means that to get an actual high quality reference genome using this technology, you need a lot more coverage than what you would need using Illumina. And since Nanopore is even more error prone, yeah, you would need a lot more coverage than what you would need for PacBio. And that is a lot more coverage than you would need for Illumina. So. Oh, yeah. Do you know what I mean by coverage? Yeah, by coverage I mean the amount of copies of DNA that we need to actually get a reliable consensus after aligning them. All right. So, yeah.
Is there any correlation between the read length and
Error percentage? Well, it does look like it, yes. It does look like there is an error, because the longer the read, that you're getting all the technology that produces the longest reads is the one that produces also the highest error rate. I'm sorry. And yeah, this goes to the length of this nanopore sequencing. The current record for nanopore sequencing, it's over 1 million base pairs in one single read. 1 million base pairs in one single read. And just a few months ago, it's been reported that the first human Y chromosome has been assembled using only nanopore data, and there are no gaps in that Y chromosome. So it goes from telomere to telomere, yep, and there are no gaps in it. There are probably 1,000 misassemblies and 1,000 errors, yeah, but the gaps, there are no gaps. Yeah, which is something remarkable. This is the first time you have a complete human genome, a human chromosome, yeah, in one single country. So yeah, that's a remarkable achievement, and they did it with Nanopore. They've also recently released some free Nanopore data, yeah, to repeat that experiment by yourselves. So if you go to the Nanopore site, you will be able to download the raw data that was used to produce that assembly of the Y chromosome, a gapless assembly of the Y chromosome.
You mentioned that it was also a chromosome, completely chromosome completely assembled.
Yep. Oh, that was a combination of several. Yeah, they started with different Illumina library sizes. So they had 300, 500, 700, and 1,000. Then they also had major libraries that were 5,000, 10,000. And then they also did PacBio. They didn't do Nanopore, but they did 10x reads. And after that, they did Hi-C. And after that, they did optical mapping. Yeah. And with all that information, they got, I think, 1 or 2 full. Yes. The human chromosome, the particularity of this, the human Y chromosome, is that it was done using exclusively nanopore data, nothing else. Yes? One contig. Yeah. The difference, we're going to touch that later, but the difference between a scaffold and a contig is that the contig, you have no gaps. In a contig, you don't have any gaps. You know the sequence from start to end, and every single spot has a nucleotide there. Yeah, in a scaffold, what you've done is you've used some paired-end information or matrix information, like long-distance information, to know that two contigs are close to each other and are probably in certain direction. So you fill in the gap between the contigs with ends. So a scaffold looks like a contig, but it has a gap or a few gaps inside of them. That's because you haven't sequenced that part. You're just assuming, you're just guessing the distance between different contigs and merging them. More errors? They do a lot of hypothesis work in this part. There's a lot of guesswork involved. They do by the guy, the director of the company in Nanopore. He said that they use different. They have at least 15 or 20 different enzymes or different pores that they test all the time, and they keep creating new ones. Yeah. The ideal part, or the most important part, is of course the part that interacts directly with the DNA. So what they try to do is try to make it either as short as possible. Why? Because the longer it is, the more nucleotides are going to be inside of the pore, yeah. While you're measuring the difference of you're measuring the changes of the voltage. So if you have 3, 4, 5, 6 nucleotides affecting the voltage at the same time, yeah, it doesn't really matter which one it just came in and which one just came out. It's going to be very complex. And the current technology, I think, it does 2 readings at the same time, or two readings at the same time, because the pore narrows at the beginning and narrows at the end, and it's wide in the middle, so that the nucleotides that are in the middle, they are not really affecting the conformation of the pore, and therefore they are not changing the voltage. But the ones that are on top, on the bottom, on the top, yeah, they are affecting the voltage. So that means that as the DNA moves along, yeah, the ones that were the nucleotides that were on the top will now pass in the bottom. So they are read twice. Yeah, that's the one they say they are really working on, and that's going to be like the next chemistry they're going to release. But that's from the last webinar they had. So I don't know if that's actually happened already or not. So now we have a clear idea of what kind of technologies there are for genome sequencing. What are the pros and the cons? Do we have any questions so far?
I hear about
Controls.
Yeah,
All right. Would you mind to? Personally, I've never done it. But what do you mean by controls? What are you trying to control? Fix? Oh, 5x of the virus. Yep. Oh, but that's for I think what you're talking about is not so much about the seed, but about the machine, just to make sure that the machine is working. For sequencing. I would lie to you if I told you that I'm sure about it. Maybe it is possible that they use some internal controls just to measure if the machine is working correctly. But if they do use any controls during sequencing of a whole genome shotgun project, they don't give it to you as FASTQ data, and we don't need it. For bioinformatics analysis, once you receive the FASTQ data, yeah, we do not need a control for the de novo. To be honest, I've never made a library. A library for this, I've never actually gone to the lab and done it. Yeah. Yeah, but there are bioinformatics approaches to actually tackle contamination if you have some. Sometimes you do, yeah, sometimes you do. But there are many ways to actually see that, and then you can claim, hey, because you usually send it away for sequencing, right? And you can just claim, hey, I send you monkey DNA, and I'm getting octopus. Why, right?
Actually, if you have a good sequencing
Provider?
Yeah, then they would check that your DNA is of high quality
Before sending you the results. But you still do the sanity check anyway.
Yeah.
I always double check it. Yep. Yeah. Mm-hmm. So, from that point of view, I would say that the ploidy is not so much a problem as the heterozygosity. Heterozygosity. Do we all know what heterozygosity means? Heterozygosity. Heterozygosidad. So basically, what we're saying is that when we do genome assembly, and that's we're gonna go deeper into this during the algorithms. When we do genome sequencing, what we're basically trying to do is to get a sample of each loci in the genome, right? That's what we're trying to do. And we don't get just one sample, we get a lot of samples from each loci in the genome. Enough samples so that we can reconstruct those loci. Yeah, that's the main idea, right? So what happens? Wait, I got lost. So what was the question I just got lost. Heterozygosity, yes. So, what happens if two loci, homologous loci, yeah, they are homozygous. It doesn't matter which loci or which homologous loci you sample. You're going to get the exact same sequence. The reads you're going to get from both loci are going to be the exact same sequence. Yeah? But if you have heterozygosity, yeah, and you sample the same loci of a heterozygous locus, what you're going to do is each of the reads is going to be different. So when you start analyzing that and you try to rebuild the path, yeah, you'll see that, hey, these look similar, yeah, but they aren't. There are a few differences, even though they look pretty much similar. So what does that do to the assembler? It confuses it. It confuses the assembly. And not only that, because of the methods that we use, which is more often than not, it's debridging grafts. Yeah, the debridging grafts use k-mers. They don't use full reads. They use fractions of reads in the reassembly process or in the assembly process. Yeah, and by using fractions of reads, yeah, basically what you do is you expect certain coverage. If you have a heterozygous region, yeah, and it produces different reads and therefore different k-mers from each of the reads, yeah, the coverage per locus will decrease because half of the reads will produce one of the homologous regions, one of the homologous locus, and the other half of the reads will be from the other homologous locus, which are in sequence, they are different. Even though they belong to the same locus, they are the same locus, they are different in sequence. So the coverage of those k-mers that belong to the heterozygous region will drop. And you will see that when we do the k-mer analysis, the k-mer analysis of your raw reads. You will see that whenever you have heterozygosity, high heterozygosity, yeah, you will always have two peaks. A high peak that corresponds to the homozygous region of the genome, and a lower peak which corresponds to the heterozygous part of the region. In most of the plants, we usually try to get homozygous plants, or as homozygous as possible. That's why we try to use double monoploids or double haploids. We try to use recombinant inbred lines. We try to use neuroisogenic lines. So we do our best to reduce the heterozygosity in our crops. There are many reasons. There are agronomic reasons for that, so that we make it stable over generations. But that has been useful to tackle whole genomes shotgun programs and avoid that heterozygosity. And that heterozygosity is actually a problem in many animal genome projects, because animals are not so easy to make homozygous. But we can see that later. Now we go, do we have a break at some point? Or do we? 10 minutes. All right, so I can just move on. So that takes us to assembly algorithms. And I cannot go deep enough into each of the details behind the assembly algorithm, but I'm going to mention the three main algorithms that dominate the genome assembly landscape. The first one, the traditional one, the one that was used for all these hierarchical genome assembly, the one where you clone, subclone, sequence, etc., is the overlap layout consensus paradigm. The overlap layout consensus, what it does, it compares. The first step is to compare every single read that you produce against every other read that you produce. It is very time consuming. It is memory consuming. It takes a very, very long time. Yeah, because you need to record the, let's call it genetic distance, or you need to record the similarity between every pair of sequences that you have. And as you can imagine, this calculation, the calculation of this similarity, yeah, the more sequences you increase, it's not going to increase, the number of comparisons is not going to increase linearly. It's going to increase exponentially. So that means that every time you add one more read to this network of comparisons, yeah, your time is going to increase exponentially. So what happens when you have millions of reads that you have already produced for your sequencing? How long do you think that's going to take to finish the overlap part. Let's assume you finish the overlap part, yeah, and you have similarity scores for each pair of reads that you've sequenced. Then you have a very messy outcome. You have a very messy graph. Why? Because some parts are going to be repetitive. Some parts you cannot use a very strict overlap alignment tool because you have to allow for sequencing errors. You have also to allow for certain heterozygosity, yeah, because that happens in real life, and that's something you need to be aware of. Therefore, some of the reads will have hundreds, if not thousands, of connections, equally good connections. How do you solve that graph? That's the part of the layout. You try to discover what's the path that makes the most sense. And this is where, well, these are the two steps where all the OLC assemblers differ. They use either different similarity matrices, or they use a different way of solving the graph that generates from having that similarity matrix. Some guys try to improve the overlap state so that you can run the comparisons faster. Yeah, that's good, but you can get a much messier graph in the end, so the layout won't work. Some people just focus on the layout, yeah, trying to order all the comparisons to make sense and to reconstruct one single contiguous sequence. It is good, but it takes a lot more time or a lot more memory. Finally, the consensus is something that most of the assemblers do agree on. Yeah, they usually use the same, or just very few. There are very few algorithms for the consensus calculation, and you could argue that the most reasonable one is the majority one. If you have 10 reads all aligned together in the same position, you have 9 A's and 1 T, you can assume that the T is wrong and the A's are correct, right? It's a sequencing error. Yeah, but that's the consensus part. The other two parts, those are the most difficult ones. This was done mostly for Sanger sequencing. This was the idea before for Sanger sequencing, because the reads were longer. And they said, yeah, this could work. Because the longer the reads, the fewer reads you need. And Sanger had enough quality, had good quality, so that you didn't have to worry that much about sequencing errors. And for a while, when Illumina was dominating the landscape of sequencing, yeah, it just fell out of fashion, because the overlap layout consensus, it's not useful for hundreds or for thousands or millions of reads at the same time. So there was another approach that actually picked up steam, which was the divergent graph, which I'm going to talk about in a second. But now that the longer reads have come back to bite, yeah, you have nanopores, you have PacBio, and they are becoming better and they're becoming cheaper. Yeah, now you have bigger reads, and the overlap layout consensus is becoming fashionable again. We have a good example of that with several of the assemblers for PacBio data. The H-gap process, for example, it uses a modification of the overlap layout consensus, where you select first, after doing very deep sequencing, you select only like 20x coverage of the longest sequences. Yeah, you use the smaller sequences to align to those larger sequences, which are going to be the seeds, yeah, and to polish them. By polishing them, you are increasing the quality of those seeds. And by increasing the quality or the accuracy of those reads, yeah, you are able to find better alignments between them. And you're not using as many reads as you would in Illumina. So the overlap step is not going to take as long. You also have a lot more sequencing to find better alignments. So the overlap stage is not going to take long. The layout part is also going to be somewhat simplified, because the main problem in the layout stage are repeats. If a repeat is longer than the read length, you simply cannot solve it, because one read is going to fall in the middle of the repeat, and you will not have non-repetitive reads from around the repeat to say, okay, let's anchor it to this non-repetitive region. You won't, because the repeat is too long. And that's usually the case, the problem with short reads like Illumina. But with longer reads, yeah, the number of sequences or the number of regions in the genome that we can consider unsolvable repeats decreases, and decreases dramatically. Yeah, so most of the repeats can be solved, and therefore the layout part, the part where you order all the reads that you've managed to align to each other into something more sensitive, yeah, becomes easier. So this will take a lot less time. And finally, consensus, we already agree that the consensus is, I would guess, the simplest part of it. Yeah. So this is the age gap process, and they've refined this age gap process, and they developed another assembler called Canoe, which follows pretty much the same approach. And then after that, they even improved it, and they created Falcon, which improves on top of Canoe and generates better, faster overlaps algorithms. And then now they've created the Falcon zip, yeah, which actually is able to differentiate between, or maybe basically to phase a genome assembly. And what do I mean by phasing? It's the same thing I mean when you genotype something. It's like you can tell which haplotypes belong to each of the parental chromosomes, to each of the homologous chromosomes. Is that a clear idea? Maybe I'll touch it in a little bit. But essentially, you would be able to tell which combination of genotypes in your genome, yeah, belong to the same molecule, to the same chromosome. Remember that you have two chromosomes, one from your father and one from your mother. And when you do small sequencing, you usually cannot tell which combination belongs to each. Yeah, but using these longer reads and using the algorithm developed by PacBio, Falcon zip, you can reconstruct it. Now there is some additional data from 10x chromium sequencing and also from Hi-C that actually helps you also to discover what are the haplotype blocks in your genome. But I don't think we're going to have time to see that, but I'll send you a few papers regarding that as well. The debridging graph, this beautiful graph. Basically, the debridging graph is a way to simplify the problem of having millions of reads and not wanting to do the comparison of each pair of them, because that's something you need to avoid. You cannot just compare millions and millions of reads against each other and generate a similarity matrix. So what they do, yeah, is they break down bioinformatically, not physically, they break down each of the reads into what they call k-mers. A k-mer is just a sequence of DNA that has a certain length. Who decides what length is appropriate? Everybody using the assembler. You tell the assembler, I want to use this k-mer size. Usually it's better if you use bigger k-mers. But the bigger the k-mer you use, the more RAM it will take, that is, the more memory it will take, because you will have a lot more combination of nucleotides to store in memory. And that's exactly why they were trying to chop down these reads into k-mers in the first place. Because if you store in memory, and this just comes down to computation, to programming, is if you don't want to compare them, then you want to do a fast lookup, yet the fastest lookup you can, yeah, to see if two sequences share the same k-mer. So what you do is you save like a database of all the k-mers that you've already seen. And you also, but by doing that, you forget, basically the computer, the algorithm forgets where each k-mer came from. So it forgets that this read had this k-mer. The only thing it cares about is which k-mers appear in my entire database, yeah, and which k-mers are more alike. So which k-mers could go one after the other. That way you don't have to compute the similarity of all of them at the same time. No, you just decompose all your reads into k-mers, yeah, assign the k-mers to the graph, basically to edges and vertex. I wish it was simple. Yeah. Let's assume here, it's more practical with an exercise. I should have put an exercise here, but let's assume that you used to have like a 100 base pair sequence read, yeah, and you decide that you want to build it from trimers, that is, sequences of only 3 base pairs. Yeah, so you start seeing in your data, you see TAC, ACG, all these trimers, and you want to see what's the overlap, which of these trimers, yeah, actually belong close to each other or just are connected by sequence. For example, TAC has an overlap of AC with this second one, the ACG. So you could argue that the original sequence here, it was TACG. There's an overlap between this node and this node. Yep. You're not actually comparing the original sequences between each other. You're not doing that pairwise comparison. You're just looking at the k-mers, yeah, and seeing in the network that it's being generated as you chop the reads into k-mers, yeah, what are the connections that you start seeing? It gets really messy, yes. It gets really messy. The shorter the k-mers, yeah, the more connections that you're going to find. And the more connections that you're going to find, well, the more difficult the layout in the end. The more difficult to solve the actual path, find the actual path. You have to choose the appropriate k-mer size. And there are a bunch of techniques to choose the appropriate k-mer size. That's why we do a k-mer analysis before we start with the assembly. Because we can predict which k-mer size will be more favorable to get more contiguous assembly. We can predict that. I'm going to leave this here because I think we're on time. Is it ready? Everything on time? Yeah.